Yeah, thanks.
All right, so our next speaker is Kenneth McGuinness. Kenneth is a postdoc, again, with the NASA project, and has worked on several collaborations between the three themes. And right now, the project he's going to be presenting is one of our connections between the first and the third theme, thinking about bridging that particular gap. It's preliminary work, but I thought that it was worthwhile presenting because I think it'll stimulate a lot of discussion. All right, Kenneth, go ahead.
Good afternoon, everybody. I am thrilled to be here. I think that this is, I kind of like the approach of having this online. It's great to see everyone from, the areas that would travel far. Y'all have beautiful faces. So today, as Zick said, it's preliminary work, but I think it's pretty awesome what these three teams are trying to do. And today, what I want to do is give you a taste of how we're thinking about connecting the mineral world, which is theme 3, with the protein world, which is theme 1. And I was thinking, about completing the Earth's electron transfer chain, and I think that's a provocative title that we'd like to get Paul's thinking about on that. So let me go through. So you've seen this slide a few times, and so what I want to focus on this slide is looking at this peptide origin world and the idea of the coevolution of mineral availability and the protein function and binding of the available metals that are around due to chemical weathering of the rocks or other resources. Specifically, what I'd like to do is take you on a structural journey for a couple of slides and tell you about my research questions. So I have 3 research questions. So one is, does an increasingly oxidizing planet over geological time affect metal coordination in minerals and proteins? So that goes back to the previous slide about we would expect the early Earth to be reducing, and then as the planet becomes more oxidized, we want to study the coevolution of minerals and proteins based on that. So a structural evolutionary connection that we're looking for is metal coordination, both within proteins and within minerals. And the third part, brings home the in-between geology and biology. What's the chemistry going on between the two of them? And that's where the completion of the electron train from the Earth to its inhabitants, we would like to uncover those links. So in the literature, it's been talked about that there's instances and there are examples of minerals and proteins that have similar geometries and elemental composition of their structural makeup. And this is, right here, you have this corresponds to right here in the Machinawatt, and you have this crystal structure of CWH corresponds to this nickel substituted gregite. And then you have this ACS active site, which is corresponding to these atoms, which slightly resembles an elemental composition, as well as the box with a couple of substitutions, the volarite. And what I'm interested in is taking this quantitatively. How do we compare them using statistics and get a solid foundation of analysis? And combing through the literature, I found ferredoxin gregite has been looked at as if they're so similar that how could these how could it not be derived from gregite, the iron sulfur cluster in ferredoxin or protoferredoxin. So looking at the structural, elemental, and electrochemical similarities in ferredoxins and gregite, so one parameter that, for instance, in ferredoxin, we see this reduced form is actually stretched along a particular axis of the unit cell of the 4-iron sulfur cluster relative to the gregite. So that kind of gives us an idea of one parameter to look at. So other parameters within the common language of proteins and minerals that we're trying to define would be bond lengths, distribution between proteins and minerals, angles, so coordination geometry compared to ideal structures. So you have tetrahedral within both minerals and proteins. So how, if you take an ideal tetrahedral structure, what would be, say, the RMSD of the geometry? And looking at the immediate coordination sphere, the elemental composition, how do they differ or how are they the same? And within the metal ligand, the oxidation states that can be found. And to bridge the gap between the living and non-living, the reduction potential, redox potential, and the band gap of minerals, the electron transfer. Basically, how do the minerals transfer energy to the proteins that Nathan spoke about yesterday in terms of these large cytochrome complexes that retrieve energy from minerals. So with that in mind, I just want to give you a taste of the data that that I've been working with. I work with Shauna and Eli, as well as Vic and Bob, on this in terms of looking at two parameters, the exact geometrical and elemental composition of metal coordination sites of proteins, which are on the top, and minerals are on the bottom. So this is a bipartite graph where the edges are the comparison having the identity where they have the exact geometry and exact elemental composition. And as you can see, similar to like arts of work that Nathan proudly discussed yesterday, I'm proud of this thing that this network kind of posted on the wall, and maybe Jan as well. She likes posting networks on walls. We've done many of those before. So as sort of an example of what this is, so basically you have on the bottom gregite, and then that corresponds to the iron sulfur cluster in ferredoxin. And one thing I want to point out here is there are many data points in this, and this parameter that I'm looking at and comparing may not be unique. So what I'd like to do is tell you about some of the work that my colleagues in Vic's lab have been working on, as well as tying in Shauna's work with minerals, and how hopefully we can connect those so that we really strengthen the bond between theme 1 and theme 3. So Hagai Renan, he took upon himself to understand the components of electron transfer and how he's able to compare the structures similar to what Sarosh just shared, the individual entities surrounding each of the metal geometries, and looked at those spheres and looked at the similarity. And then what they did was they added another element to it. They went on the hypothesis and the observed information that in order for electron transfer to occur, they have to be within about 14 angstroms within the structure. So that's where we get the span idea, spatial adjacency network. You have these structures that are similar in their their coordination, similar to what Jana was talking about when you align them, where the distance of the metal cofactor is the base for how to determine whether or not an alignment is true or not. But you also have this factor of electron transfer where they have to be within a certain distance from each other within that protein. So you have the spans, for instance, the 1 are structures that are similar, and then the edges are, they're close enough that electrons can pass from one to another. So the beautiful part of this data set, and Eli did this work with Hagai and Doug, and that what they saw is that they were able to map on the perceived metabolic pathways throughout deep time onto that spatial adjacency network. So they're reducing the data. So that theme of reducing the data of what I showed you before of the connections within the to the minerals to the proteins is my aim. And these are the tools that I will begin to use in addition to the work by Shona. And Shona put out a beautiful paper in 2017 highlighting how network analysis can be applied to mineralogical systems. And the beauty of this work is that she combined time, which is inherent within geology, being able to date minerals and rocks, with co-localization of those minerals to understand more about the geology and the evolution of mineralogy systems. So what I want to do is actually connect these two. And the connection is such that we get time in both components. We get a geological time estimate, but we also get a metabolic estimate based on the biosignatures within the geology. And what I expect is that we're going to use co-localization. Adrian and Jiwa and I've been talking about organisms that are within or surrounding those minerals on Earth. Where are they located on Earth, and seeing the metabolisms that are involved in there and the structures, and seeing how those can be also connected on another level. So there's a lot of tools that are available for the connection between theme 1 and theme 3 that will enable this reduction in data. So speaking of Eli, so this is the questions. The first question that I'm asking is, how did the change from a reducing atmosphere to an oxidizing atmosphere not only affect mineralogical evolution, but also affected mineralogical evolution's interaction with the growing biosphere that it helped create, as well as the biosphere then interacting and modifying the geosphere. And you can see here that you have metabolic invention with confidence around loosely 3 billion years ago, when we have oxygenic photosynthesis. And then you can see here we have magnesium becomes a new metal that is still going on, and then manganese, which is a not seen in other pathways. So that gives us evidence that it seems to be the case that it is changing the metabolism of the organisms based on the available minerals or metals, at least. So getting to the crux of the title of my talk, completing the Earth's electron transfer chain, and this gets at looking at the redox potentials of proteins, but also looking at the band gap, or the ability to transfer electrons within minerals or solid material. And this is something that I'd like to talk to Shauna and Bob and and others who may know of resources to calculate, or maybe there's databases to understand what, or band gap of minerals. But this is the interface of the discussion of energy transfer I'm imagining to occur, where you have a guy's most recent work where they're able to look at the span in a new light, where they can understand ferredoxins in the middle, where they reduce the other entities within metabolic pathways. So then you have this expansion of reducing to oxidizing. So hopefully we can come up with this link. So with that tantalizing bit of data and the tools that I'll be using to reduce it, I'd like to thank Vic, Shauna, Paul, Eli. I also want to thank my rotation advisors. I spent a lot of time with them, and they gave me a lot of, I think Paul said that and Yana said that the vision for this NASA project was to have this not only cross-disciplinary, but also being able to do rotations in other labs and get a first-hand experience in network analysis, which I was able to do with Yana, as well as look at the mineralogical side of it with Nathan. I think those are tools that I learned in those labs are vital to connecting. As Yana said, theme 2 is the connector between theme 3, and looking at networks and how to connect them together and what parameters we can use to do that. So a special thanks I'd like to thank Inspire, which is my funding agency through the NIH, that they are participating in this RISE program, which is the Research Intensive Summer Experience at Rutgers. And I've been blessed to have 4 incredible students this summer that I'm managing, and they're managing me. So Justin, Nolan, Isabel, and Falade, where we're going to break this task into chunk sizes, and we're going to tackle this connection between theme 1 and theme 3 using the parameters that I shared, as well as other parameters that those on the call may suggest, which I'm absolutely open for sure about your thoughts and how we can further this along. I also want to thank Shauna for her laborious work. She's been culling the data set of the crystal structures by hand, and for the minerals, and I just wrote a script to do it, but she's doing it by hand because of the importance of the data set that for culling the minerals. You want to make sure that we have the appropriate data set to work with, and she's doing a stand-up job for that. So thank you. And then everyone on theme 1, theme 2, and theme 3 that are not on this list, you guys are also mentors to me. I listen, I learn, and appreciate it. And of course, NASA, we wouldn't be here without you. And yeah, thank you.
All right, thanks, Kenneth. Any questions?
Ken, great talk. I don't have a question. I just want to make you aware of the large data set that we've been building for the past year. Me, Shauna, Donato, Bob, it includes a lot of mineral analysis. So looking at location-specific mineralogical assemblages that are in the MinDat repository. So if you and I should definitely get together and have a discussion over what you need and what we have.
Excellent. Wow, that's fantastic. Great. Yeah, there's definitely, we won't sit down in a pub and write a paper, but we'll, right, Ariel?
I want to make a quick comment that the concept of a band gap is in solid state physics. It used to be called an energy gap, but for those of you who are not aware, there is a valence band and a conduction band. So between the two, there is no possibility for an electron to exist stably. You can have an electron popped out of a valence bond into a conduction bond, and that's basically an ionization reaction that requires a lot of energy, depending on the the mineral. So that this concept is related to the redox potential in proteins. And that's, I think, exactly what Kenneth is trying to get to. I mean, he's so used to thinking about band gaps that it's not common in biochemistry to think about band gaps. But I just wanted to make sure that people understand what it is.
Thank you, Paul.